Okay. Um, Hi, everyone. Nice to see you. And I'm going to talk about just Shamini asked me to talk about general um, remarks about to provide you some perspective about what all of this data might be useful for that we're generating. So I'll talk about a little bit more general and a bit theoretical things, but also some specific interesting ideas that we can use Visium and generally spatial transcriptomics data for. So I'm a computational biology professor at U of T, and my lab develops computational methods, and we also do a lot of analysis at the Donnelly Center. I'm also part of a program called the Multiscale Human Program, run by CFAR, which is the Canadian Institute for Advanced Research. Their office is actually just on this floor of this building across the hall. And they're an interesting funding agency that funds workshops over long periods of time. Some of the programs have been going for 40 years. So we applied for a topic called Multiscale Human. So we have these workshops with a whole bunch of different people that come regularly. And our idea is just how to think about mapping the human body and understanding it from across scales, from molecules to tissues, all the way up to the whole body. Of course, that would be useful for lots of medical applications if we could, the more we learn about the body. One of the things that we thought about or discussed at these workshops is that fields that traditionally try to understand the human body have been siloed across scales historically. So molecular and cell biology fields have focused on molecules to cells, and physiology and medicine have focused on physiological level systems. And they don't really talk to each other very much. It's not very integrated, but single cell and spatial genomics creates a bridge. We're getting lots of information about multicellular systems and tissues that creates a really nice bridge between all these fields. And it raises the question of whether we can come up with a unified field that considers how the whole body works across scales. Because, well, we finally have information, a lot of rich information across scales from molecules all the way to whole body. If we can measure this information, we measure lots of different types of information about the human body. We can measure single cell transcriptomics, spatial transcriptomics. We can do whole body imaging. And we have tons of information that we've learned about the body that is published. And if we think about we think about the genome as a model that has all the parameters to generate the body. Of course, there's some environmental components of how that works, but most of the information is in the genome. And we can think, anything that we measure about the genome, anything that we measure about the body, because it's all generated from the same information in the genome, should be correlated somehow. And we can also think about an analogy of the genome as the ultimate generative model, an analogy to generative AI. So most of you know about ChatGPT, and generative AI and generally is about developing models that can generate new patterns that they learn. So ChatGPT can generate English text and any other language, and also computer programming, any kind of text-based language that has a grammar. You can also make generative models that learn images and how to generate images, and you've probably seen some of these where you can give it a prompt and it will generate a picture or a photograph of someone, which is not a real person, but it's learned how to generate that from a distribution of information. So there's, if this analogy holds, one of the nice things about generative AI is that it can self-supervise based on information that you feed it. So the way that traditional machine learning worked is that you had to spend a lot of time, people had to spend a lot of time making training data. So we'd say, okay, this is a positive example, this is a negative example, and spend many hours organizing that data. And then we tried to use machine learning we teach the machine learning algorithm the difference between positive and negative, we might say and we might try to use it to, for instance, classify a histology slide as a cancerous, tissue or non-cancerous tissue. So for that, we would have to collect lots of histology slides and manually annotate them, cancer, non-cancer, and then the machine could figure it out. Now, with generative AI, there's a new way of working, which is self-supervised machine learning. So instead of giving it those training data, you give it all those training examples, you just give it all the data and it learns. So in the case of ChatGPT, we collect all the text that we can collect, and that model was trained by trying to predict the next word. So you can take some text and you can hide a word, and you can say, based on all the rest of the text, guess what the next word is going to be, and then you know what it is because you hit it from the model, but the model doesn't know. And you can train the model to update its weights automatically with the current algorithms that are available to do really good at that. And then it turns out in that particular case, it was able, like once it did that, it was able to speak to people. So it was kind of an interesting emergent property. But the main value of it, I think, for biology is that we can generate lots of data and feed it to these machine learning systems and have them automatically learn important structures that we can then use to classify cancer a little bit more easily than we could before, for instance. So is machine learning a good approach for understanding the body? I argue yes, because evolution has created the biological system through copying and mutation operations. There's a memory and there's selection, but mostly biology is about copying and mutation. Copying generates redundancy. So every cell has a lot of information, similar information as every other cell. That's great for machine learning because it needs redundancy to help with pattern recognition. And then mutation generates variation. We know that every cell is slightly different than every other cell. There's some variation. That's really important to understand how data measurements relate to each other. If we didn't have variation, we couldn't compute correlation or causation. Just we wouldn't have any information about it. So that variation is really useful, valuable information. We still need to understand mechanism. We can't just let the machine learning system do everything in a black box for various reasons. One example reason is that if you have a mutation in an individual that's not present in other individuals, the machine learning system probably is not going to be able to understand what that mutation does. And that's an example where rare events will require some mechanism-based interpretation. We have lots of information about genetics that we can use to also understand the scale. So we know, for instance, we have a mutation that we can that it might affect a protein or complex pathway, cell, tissue, body. It causes disease, and there's lots of genetic data in biobanks. And so combining this information will help us as well. So you can think about how that relates to spatial transcriptomics data. How do mutations affect the patterns that we see in the spatial transcriptomics data? So how would we go about doing this, learning about the body at multiscale? Well, we have to collect a lot of data. There's lots of projects that are collecting data, and Visium and spatial transcriptomics is an important component. But ultimately, we would like to test that we've learned something, and we usually have some models. These days, because there's lots of different types of computational models, including AI generative models, we could use those potentially. We'd have to test that they're working well, so we'd have to test that they understand mechanism, that they're accurate at predicting non-trivial tasks like the response to perturbation, or even more difficult would be predicting information from one scale, take information from one scale and predict what's going to happen in another scale. So look at spatial gene expression data and predict what's going to happen with the whole body, for example. And if we could do that, there would be lots of useful medical applications. There's this idea of a digital twin, which is a computational model that learns, it's parameterized based on measurements that we make about an individual. And then you can do tests on the digital twin. And then you might use that information to update what you're doing in the patient, for instance. Like you might change the parameters of their treatment and then cycle over and over again to optimize it. So that's kind of just a bunch of thinking about how spatial transcriptomics fits into maybe a bigger picture of understanding the body. I mentioned in one of the earlier slides that if the body is generated from information in the genome, that all the information we measure about the body should be correlated. This project that I'll tell you about now is an example that tries to measure that and learn it automatically. This is a project by Ronald Shea, who's a PhD student in my group, co-supervised with Bo Wang, who's at the Vector Institute here, and Quan Pang, who is an undergrad student from U of T, University of Toronto, who worked on this as a summer project. So what we want to do in this project is take spatial transcriptomics, in this case it's Visium data, and try to take the H&E image and predict or learn the relationship between the H&E image and the spatial transcriptome. So, and again, if we expect that our hypothesis is that these should be correlated, and that means you should be able to predict one from the other. Okay, so how do we go in about doing this? We developed a machine learning method that takes the H&E image and the spatial transcriptome. The H&E image is split up into little patches or little blocks, and each of these images corresponds to a spot that is the transcriptome on the Visium slide. And so we take those images and we feed it through an encoder that converts it to vectors that allows us to embed it in a space that you can visualize as a UMAP, for instance, where all the dots would be little patches of images. And we do the same thing for the transcriptome spots. This is not Visium HD. This was the previous generation of Visium, but it's the same idea. And we encode that transcriptomics data as also vectors. You can do the same thing. Think about it as a UMAP, where each spot is a transcriptome spot. Each point in the UMAP is a transcriptome spot. And then we use machine learning to automatically align these, learning a joint embedding, where if two patches are similar in this space, then their corresponding transcriptome should be similar in this space. So the machine learning method tries to kind of jumble around all the parameters until it figures out a way of aligning those, and it uses something called contrastive loss. It's a deep learning method. And then once we learn this, we can convert one to the other. So we can take an H&E image patch and predict its transcriptome based on what's the pixels that are in that patch. So how did that work? So it worked reasonably well. There's multiple methods that do this now. It's a hot topic of research. This was the original Visium slide. This happens to be a human liver tissue that we got from Sonia McParland and Ian McGilvery at University Health Network here. And we visualize one gene here, CYP3A4, which is an important liver enzyme. And you can see how patchy it is across this liver slice. Just given the H&E image, this method, BLEEP, was able to predict this pattern for this gene. And if you look back and forth, you can see that the patterns are related, and it's about 74% correlated. That was the best case. We had a bunch of other genes that were pretty correlated, but on average, the correlation across for all genes was not super high. It was like 22%. That is better than the previous methods that were published at the time, but of course, it's mostly not as correlated as you might like. But it illustrates that this was pretty successful for us because it illustrated that you could automatically learn to predict one modality from the other, image to transcriptome. We could go ideally transcriptome to image as well. We didn't study that part. And since then, people have published better methods and with more data, and these numbers are going up. Kind of interesting to think, we haven't done this test, but you might think, oh, well, how well can a person do with this? If you give the H&E image to a trained pathologist or histologist, they could probably look at all the identify cell types from the image. And once you know a cell type, you would know the markers for that cell type, and you know, you can compare how well this method does versus a human expert. But probably over time, that computer method is going to be better and more generalizable. I want to mention a couple of other little things. So one interesting thing was that we want to collect more Visium data to extend this work. And the gene expression omnibus has lots of Visium data, lots of spatial transcriptomics data. When you generate your data, you'll likely submit it to a repository when you publish. And Geo is one that accepts spatial transcriptomics data. We wanted to get all the data, so Ronald was able to try to use ChatGPT to help with this. So we asked for all records that contain the mention of spatial transcriptomics, downloaded all the identifiers, automatically got all the descriptors, the descriptions from Geo for that, and then we put that through ChatGPT, and we asked it to say, well, you know, categorize all these samples. Are they human? Are they mouse? Which cancer? Which other disease are they studying? And we got that table. Here's if anyone's interested, here's the kind of prompts that we use to ChatGPT. So this includes prompt engineering, like little tricks that people have figured out help make the ChatGPT work better. You're an expert biologist, and you give it some context. What type of cancer is this from? You have to answer from the following list. Can't just make up your own things. And that helps. And then you can ask it to figure out how confident it is, or explain how confident it is, and it will tell you, you know, I'm very confident or less confident. One of the interesting things we noticed in this project is that a lot of the Visium data sets that were curated or collected from Geo did not have full resolution images. So when you load up your Visium data in the 10x genomics methods, so typically the H&E image would be imaged with a 20x or 40x microscope magnification, and you get the full image from that microscope. But when you load it into 10x genomics tools, frequently it gives you, well, it will save like a high res and a low res. That high res is not the full resolution. It's just higher than the low res. Those are optimized to make it easy to quickly view the results on your computer screen, but the full image is hidden in there and not easily up. You know, you have to dig to find it. And so most people at the time when we looked at this were uploading those low res or high res images and not the actual raw data from the microscope. And we found that if we used higher resolution data, this bleep method did much better than if we used the low resolution data. So when we share the information from spatial transcriptomics, it's important to share all of the data, not just the spatial transcriptome, but also the images. The images are very valuable. Most people use them, and I don't know what was mentioned earlier, for quality control in the experiment, but it's actually very valuable information that, if shared, can be used by other people or yourselves to do interesting things. One of the interesting applications that people are working on is to find out if spatial transcriptomics can help improve pathology decision-making. So right now, most pathologists use just H&E images, and they're able to have some performance with, they're able to reach some performance with classification ability for diseases. And spatial transcriptomics gives us a lot more information about the sample. You can't get spatial transcriptomics these days because it's too expensive for every sample. So we're going to be stuck with H&E images and pathology for a long time. But maybe you can collect enough spatial transcriptomics data to learn the relationship between the H&E and the transcriptome, and then update the H&E model so that it does the machine learning system, for instance, can do better than a pathologist at classifying tumor, for instance. And people are already publishing some papers that are suggesting that's possible. And you won't need to have spatial transcriptomics for every sample. You'll just need enough spatial transcriptomics data to learn how to do that task what better than a human. And then you can then just use it on H&E images. So things like there's interesting applications that we can think about that could extend the value of the spatial transcriptomics data outside of just the individual experiment that you're working on with one or a set of data. So I just wanted to go over those slides to suggest a few different ways that you can think about using spatial transcriptomics and think about it maybe more broadly, and give the important message that you should share your data, and that data, all the data that people are generating in genomics is being analyzed by lots of different people, and including by models that will collect all the data and try to make very valuable, enable very valuable applications like the clinical applications I mentioned. So so those are all the slides I want to share for the final thing. I can talk about other things if we have time, but I wondered if people had questions.